Institute of Pharmaceutical Science, NEMCARE Guwahati Division, Mirza Farooq Assam. The topic of today's FDP program is Basic Statistical Techniques Used in Health and Clinical Research. I also want to welcome all the participants who have joined today with us via online mode. I feel you all will feel enriched with knowledge after completion of this great event. Today, we have the chairperson of today's FDP, Dr. Bishwa Ranjan Phukan sir, Academic Registrar, ASTU, Guwahati, Assam. We are very much privileged to have our respected convener, Dr. Bhargav Jyoti Sohoria sir, Principal NIPS, and Organizing Secretary, Dr. Apurva Talukdar sir, Vice Principal NIPS. We are also delighted to have Mr. Nalini Ranjan Sarma sir, Principal Saul Brook Junior College, Mirza, and Mr. Bikram J. Kashyap, Administration Head, NGI. Our guest of honor for today's FDP is Dr. Monita Baruah Madam, Department of Statistics, School of Mathematics, University of Leeds, United Kingdom. At the meantime, I would like to read a brief bio of Madam. Ma'am pursued her BSc and MSc degree from Cotton College in the year 1993 and 1996 respectively. She was awarded PhD from Brunel University, West London, United Kingdom in the year of 2007. Previously, she was working as a lecturer in statistics from January 1999 to December 2010 in Department of Statistics, Cotton College, Guwahati, and also as a lecturer in the Department of Mathematics, College of Science, University of Bahrain from February 2011 to January 2020. Courses taught by Madam were like mathematical and applied statistics at undergraduate and postgraduate level, mathematical statistics for engineering and IT in the undergraduate program, queuing theory for MSc, etc. MEM has various teaching and mentoring skills. MEM is the recipient of gold medal for securing first class first position in MSc in statistics from Guwahati University, Assam, India. MEM has various publications in renowned journals and also attended various seminars and workshops. MEM is a member of professional bodies like All India Association of Educational Research, Bhubaneswar, Orissa, India. Presently, MEM is working as teaching fellow in statistics since February 2020 onwards in School of Mathematics, University of Leeds, Leeds, United Kingdom. So we all welcome you, ma'am, and we are also very grateful that you have given us your valuable time, and we all are looking forward for a great program. So at the meantime, we'd like to move forward for the felicitation program. So I would like to request our honorable principal sir to come over the dais and to felicitate madam. Thank you, ma'am. I again request our honorable principal sir to felicitate Dr. Bishwa Ranjan Phukan sir. Sir, please. Now, I would like to request our convener, Dr. Bhargav Jyoti Sohoria sir, to give inaugural speech and to inaugurate the program.
Thank you, Deepsika. Respected dignitaries present here, all the faculty members. Today we are very happy and pleased to organize such a program for faculty development and mathematics. Science without mathematics is zero. Proverb is there, and faculties are the petals of a flower. And if the institute is a flower, without the development of the petals, institute cannot be bloom. The fragrance, if we petals will be developed, we will like very beautiful flowers. If the flowers is very beautiful, it provide the fragrance to the society. So that is why from the beginning we are trying to develop our faculties. We are trying to develop our petals such that we can bloom in the society. And we are very happy that Dr. Monita Baruah ma'am is with us. Definitely, NEMCARE group of institute from the beginning always try to offer a tribune to the researchers, always give a tribune to the scholars, always try to offer a tribune to the human resource. Because we are the responsible person to bring the best human resource to the society and to develop our society. That is why to develop the society, the faculty should be developed. That is why we are organizing a faculty development program here. And we are very happy that Assam Science and Technology University from the beginning is always trying to collaborate for these different events and always trying to encourage us, our faculties, to develop our societies. Thank you very much. We welcome you, ma'am and sir, to NEMCARE Group of Institution. Thank you.
Thank you very much, sir. Now, I would like to request our respected chairperson of FDP and academic register, sir, Dr. Bishwaranjan Phukan, sir, to say a few words with respect to this event. So.
Thank you so much. Azir MDP Khonor Mukho Otiti Sabi Asolta মই ক'ব লাগে যদিও চেয়াৰমেন কৈছে মুখ্য অতিথি মই মেডামকে ক'ব খোজো গেষ্ট অফ অনাৰ টেক্টৰ কৈ থোৱা হৈছে she is the most important personality today in this evening MDP and yes Dr. Bhargav already mentioned that we need to develop ourselves with a dynamic world. Every time, in every fraction of seconds, the science are changing. The standard are also changing. What was the standard yesterday is not today. So, and the system also very complicated. Particularly the pharmacy related to the pharmacy, which play with the human or this biological biology. I mean, with the live animal or the human being. It's a very difficult. Our system also sometimes responds to some drugs is nicely, sometimes reversely. That still we do not know. And that's require a lot of examination, lot of experiment, etc. And sometimes it's become very difficult to draw the decision. Yes, we have the tool through the statistics, mathematics, which help us to draw the decision in this, particularly in the type of and the situation of complicated situations. So the statistics and mathematics and these are the tool. And biostatistics and bioinformatics are more specialized in nature that to help our you can say that to support our decision-making process. And I'm very happy to be here today in this FDP, Faculty Development Program, that will certainly help our young faculty who have just entered to their scientific world for research particularly. That will help a lot, and I really thankful to Madam Baruah, particularly for her sparing time to be here and in person and to interact with our young faculty of these institutions, and also the faculty from the other places from Assam and outside of the Assam also. Assam Science and Technology University, I assured you from as being an academic registrar, will try our best to provide maximum support to our colleges. Yes, sometimes we have some difficulty, of course. Sometimes maybe because of the manpower or because of some other difficulty we have, but as a whole, we are trying to support all the institutions of our possible best. And sure, the faculty, the better faculty, and will help growing the university. So this is very specialized in nature, and the use of statistics, particularly in the human and animal world, you can say. And I also extend my thanks to the NEMCARE group, and in person Dr. Bhargav and his team for organizing this event, and also helped you can say the association with the Assam Science and Technology University. I expect a great day and great learning, particularly from the madam, and this is the just beginning, we can say that. And all the faculty member, I hope, can have their relation with the madam through email or maybe over telephone when they find the difficulties in their research or in their teaching. Thank you so much. Thanks to all for giving me this opportunity to be here today. Thank you.
Thank you very much, sir, for your wonderful speech. Now, without any further delay, we would like to move forward for the main program. So I'd like to request Dr. Monita Baruah, madam, to please come over the dais and deliver her speech. Okay. Okay, good afternoon, everyone. Good afternoon. Thank you very much. I'm honored today. Thank you, respected Chairperson, Academic Registrar, Sir Sharanjan Phukan, Dr. Bhargav Saharia, Dr. Purbo, title to my and Nilutpal Vikramjit, everybody of the NETS group. I'm very much honored to be here and thanking you again for giving me this opportunity. Start কৰি নিদিও মোৰ নামটো গম পালে সবে মনিতা বৰুৱা statistics বুলি ক'লে এটা ডাঙৰ এৰিয়া হয় Let me just start my introduction just by giving you some glimpses, some snaps of the university where I stay. This is the University of Leeds. It is a very heritage old university. It was established in 1874. This building is the landmark of the University of Leeds. It's called the Parkinson's Building, which was named after Frank Parkinson. He was an electrical engineer, and he has a lot of contribution to this university, to the growth of this university. So it is a library hoy, and there are facilities as well. So the University of Leeds, Google search korile, it dekhiba. Then this is the department of, or the School of Mathematics. We call it the School of Mathematics, and there are different departments under that, like Department of Statistics, Department of Financial Mathematics, Department of Pure Mathematics, and so on. Saidaman Mathematics or Department, and this is the Great Hall where the convocation ceremonies takes place. So Tinta was to my required abruptly start. So this is a beautiful place, and it's just been a 1 year. I have started working there, and I feel privileged that I'm a part of that university. Moving on now, so let us start with our FDP program. That is my topic of my presentation today is basic statistical techniques used in health and clinical research. Okay. I'll just have an agenda. I'm not going to stick fixed according to this outline, but just to have a grips of what I'm going to speak today. So it's just been an introduction and something about basic and scientific research, role of statistics in health and clinical research, statistical techniques and methods, and then we'll have a question and answer session or interaction session. Undoubtedly, you have noticed that there has been a lot of progress in the medical and the health sector. Over the years, I mean, there have been a lot of progress. And without any doubt, this has been due to research in that area. So much of research that now these cancers are being treated if it's detected in the early stages. So without research, I think it's not possible. There was a low mortality rate, now there's a high mortality rate. People are living, the life expectancy has increased. So these are due to the advancements in research. Right? So what does statistics do there? Yes, statistics and research, especially in the health sector, that's the epidemiological sector, has a large history. It's been since ages. The father of field epidemiology, John Snow, in 1854 studied the cholera epidemic in London and demonstrated the association of epidemiological and statistical methods in medical research. Statistics has been used since then. So for any kind of medical or clinical research, you have to apply the techniques or the tools of statistical research, statistical methods. So I think most of the faculty members are either researchers already or have a doctorate degree, or they might be ongoing, having a research just going on, or maybe are thinking about doing research. So I think it's very important that you know the basics of statistics here. So when we think about research, what comes in our mind? What do we want to study? What are we going to focus on? What is the problem of study? What is the purpose? What is the objective of doing that study? What is the appropriate study design? So there definitely has to be a design to study, to answer that problem, whatever you are willing to study. And what appropriate tools and techniques are required for the study? So what kind of study you are going to do? You have to apply some tools, some techniques. So this is what, in a small nutshell, research is all about. And then we broadly classify, or we just divide research into two main categories. That is the basic research and the scientific research. Basic research is if you want to know something, if you want to have a knowledge, advancement of knowledge, you know. So basic research, also called fundamental or pure research, has its primary objective is the advancement of knowledge. You want to know something more. And the theoretical understanding of the relationship among the variables. Variables in the sense that
What are you trying to study? So that's basically, suppose I want to take an example. How has COVID-19 affected the hospitality industry? Hospitality industry, COVID-19 amar hotels bor, hey sector book and affect. So that's our inquisitiveness, a curiosity to know how it has been. So that's also one kind of research. So that's fundamental research. So it is exploratory and often driven by the researcher's curiosity. So you want to know something more. Interest and intuition. Therefore, it is sometimes conducted, you don't have an intention in mind, but you just want to know that without any practical end in mind, although it may have some unexpected results. We might take some actions on that. So that's the basic research. If you want to do further study, we do it on the basis of scientific research. Oh, sorry, I have an example here, I forgot that. So the research question can be, what I already exampled you how COVID-19 has affected the hospitality industry. So there can be another question that, what are the causes or the risk factors of prevalence of a disease? So at a disease, so what are the factors? So we want to enrich ourselves, we want to know more, we want to explore. So that's our basic research. So that's just an example of a research question. Then scientific research. Scientific research has certain rules. We have to follow certain procedures. So generally, research is understood to follow a certain structural process. These are formation of the topic. You have to know which topic are you going to handle. Then we have to have a clear hypothesis that this is what we are going to study. Then when you're going to study something, you definitely need some information. We call it data in statistical terms, or we call it numerical information. So we collect the data and we try to analyze the data, and we do some kind of testing. Then we go to the conclusion, or we say inference. In statistical terminology, we call it inference. So this is the basic structure of scientific research. So the research questions, scientific research, how well a drug or device works under ideal conditions. So we want to look at the efficacy of a drug or a device. So how is the MRI machine, sophisticated MRI machine, working under ideal conditions? So we might have, that's one kind of a research. This is just an example. My medical field or not, I'm just trying to give some examples on that. Then how well a vaccine is effective in the immunization process. I think this is a very burning research topic. Most of the researchers are working in this area because of the COVID-19 vaccine. So there have been trials, vaccine has started. So how has been, how effective has it been? In fact, I'm also thinking of doing something on that. So interest you can take Assam as an area or Guwahati as an area and think about that. Then application of statistical model to patient flow in emergency department. Hospital administration statistics. So you can apply a model to see the patient flow, depending on how the patient flow and how we can take important decisions. So that's some questions, examples of some questions on scientific research. And the process of research can be quantitative as well as qualitative. It depends on what area you are looking at. So sometimes there is quantitative and sometimes it is qualitative. And the outcomes, the results of the research can be applications. You have to apply it, but it can be just for knowledge, basic or pure, or some decisions can be taken. So it's an action. So that's the outcome of the research. This is just to give you an idea on the research, how you can start doing that. Now, biostatistics has developed enormously in recent years. Everywhere we need statistics. So in diverse biomedical areas and fields. Clinical trials are an indispensable part of the medical research because through clinical trials, scientific finding can lead to improved ways of preventing, detecting, and treating diseases and clinical scenarios. So this is, we already know, but statistics here acts as a critical part at all stages. So starting from data collection to testing, a statistic has its role. from planning to conduct and interim analysis to final analysis, and then providing a report. And due to the different problems arising in biomedical research, there has been also advancements in statistics. So, jimane complicated issues ahi gose, there our statistics have simultaneously being developed. Our notun techniques we have started discovering and knowing about that. So new problems in biomedical research have led to the development of new statistical methodologies that would not have arisen have if biomedical area to the progress in hole hai. So what is the role of statistics in now, when I say statistics, it's a very vast area. It's a very vast area. So in this presentation, I cannot dedicate everything. So I'm just looking at certain points, certain topics only. So just a common overview I did develop, because if you want to really learn statistics for use as a research, you can take some courses or you can consult a statistician, but I can just give you an overview of if you want to really learn in depth, then you can do more studies. Okay. So statistics as in medical research or health research starts at the initial stage of a clinical trial or a laboratory experiment. While making inferences, inferences is the conclusion concerning a wider population. Always we, whatever conclusion we will do, we will speak about the whole population, not about the sample. Population is generic here. Population does not only means humans. It can be anything here. So the wider population. And analyzing a large data with lots of information, statistics is important. In medical field, a lot of uncertainties there, a lot of variables. Always keep something changes. There's always changes happening in medical science. Even in COVID-19, we have seen in UK there's a new strain of COVID-19. There's a new mutation. So severity to Baruah. So that's always a change. Now researchers are trying to think about detecting how it can be. Is it more fatal? Is it more, is the mortality rate increasing due to the new strain? So there are a lot of changes happening. So whenever there is uncertainty, there is statistical methods are helping in creating solutions for this type of complex situations. Sophisticated research projects often need advanced statistical methodologies as well as mathematical model formulation, including testing and drawing relevant conclusions from observed data. So that's how statistics fits in medical or health or clinical research. Or moving on now. Let us Monita concentrate Korim in the statistics part. Okay, that's not just giving you some idea about what is research and how statistics can help in pursuing our research. So we generally broadly classify statistics into two different types. One is the descriptive, and secondly is the inferential. And the descriptive part we usually organize. We look for patterns. We try to gain insights and be familiar with that subject, describe the data using tables or graphs, and this will also leads to further investigation if you want to do. In inferential, we use information from descriptive statistics to make decisions or predictions about a population. So Amar, we have discussed, we have done some descriptive analysis, but this will help in doing an inferential analysis later on. So that's mainly done through observational studies, case reports, and intervention or clinical trials. So these are the three types of doing inferential, using inferential statistics. So what is descriptive analysis? It refers to statistically describing, aggregating, and presenting the variables of interest or association between these variables. So variables are what we are going to study. The variables will collect into area that's variables. Descriptive analysis are likely the most simple of the statistical analysis to perform, and anybody can do a descriptive analysis, okay, and to interpret. So it's also very good to provide conclusions. Analysis of data that allows us to generalize. I mean, if you want to generalize from a sample to a larger population, okay, requires the use, then you cannot use descriptive statistics. You have to use inferential statistics. Okay, when we are going to do descriptive statistics, the first thing we need to know is we have some information. So that what is that kind of information? That's numerical information. And numerical information is the data. We have to have some data. So data, there are different types of data which we, it depends upon what are we going to study. So there's, firstly, we can say there's a qualitative or nominal variable. So measures do not have quantitative value. There is no numerical value involved. They are generally labels or names. For example, it can be a race, gender, or adverse event type. Like adverse event type means like cancer, cancerous, non-cancerous, benign, malignant. So these type of things are adverse event, moderate, severe. Then what type of summaries are standardly used for this data? So if you want to do some kind of qualitative analysis, what type of summaries we can use? We can use frequencies, counts, and percentages. So this is very simple. The next is the quantitative, which is ordinal. Sometimes quantitative data is ordinal. There are two types actually, ordinal and ratio. Measures which have an order. So again, it's an adverse event like mild, moderate, severe. There's something which is in order is adverse event. Satisfaction scale, very satisfied, unsatisfied, somewhat satisfied, or average, below average. All those types. Then what do we do in this case? Again, we use frequency, counts, and percentages. Now again, here comes mean and standard deviations. So, and along with that, median. These are measures of central tendency. The mean, standard deviation, whatever we do, summary statistics, that comes under a topic called measures of central tendency. The next type of data is the interval or a ratio, which is also continuous. This is the real life data. Wherever you see measurements, the height, weight, temperatures. So these are all real data. That we call continuous. Or the decimal points. Continuous variable. Measures have an order, and they can also have an order and exact differences between these levels. So, for example, systolic blood pressure. Many blood chemistry and hematology lab values. And how do we summarize them? We summarize them with the help of the measures of central tendency like mean and standard deviations, medians. There are more quartiles, bahut Assam. But I'm just giving you the most popularly used. So these are some of the summary measures. Which you will, I think you have always seen these in any exploratory analysis, any descriptive analysis, you have seen that. So there is the arithmetic mean, where we take the average of a set of values, then we have the standard deviation, which decides the amount of spread around the mean. Now, the standard deviation or the variance is very important. I just want to give you a small example in understanding the standard deviation, because whenever you're using data, it's very important. Suppose my medical field or not, let me talk about something very simple. No problem, thank you. Suppose okay. If you want to, there are faculties will here. So if you want to know the performance of a student in the last exam, so I mean, average marks to it was 60%. So it's not necessarily 60%. So 60% is the average. There are some students who may get 40, some students who may get 50, some students who may get 80. So you know the variability from the mean. So that's what we call variables, the distance of the values from the mean. So that's so different values are there, and we take their average. So the distance from the central value, that's our spread, and that is measured by the standard deviation. So that's very important. And then we have the median. It's the middle value, the range. I will not go to the details because already there you can have a look. Then we have the interquartile range, which also determines the amount of spread around the median. Then we have the frequency measures, which is the proportion which describes the fraction of times an event occurs to the number of times that event has happened. So that's proportion or a percentage. Then again, we have the incidence proportion, that which is risk. So if you want, you can go through this chart and understand the different tools for summarizing numerical data. So let us do a small example on, I have taken this information from the COVID-19 dashboard as of 7th January. This data is based on the 7th January. So if you look at this data, it will look actually numbers. So when you say how many of them are active cases, you have to just say, okay, there's 3,525 cases out of 21,000 to 2,15,510. So it doesn't give a good, it's not, we cannot just recollect what's actually the numbers. So if in case we want to give it, summarize them in the form of some pictures or some graphs and proportions, then it gives a much more clearer picture. So if you notice, we have seen that in the confirmed cases in different districts of Assam as well. So let us look at some charts of representing this. So if you see in this graph, now it's very much clearer. Instead of that tables, we find that, okay, now the number of active cases are very small by looking at just the graph. So this is giving us some insights, right? Okay, in this graph we say district wide, oh, Kamrup Metro is doing very badly. They have a high number of cases. So this is what statistics is for. It just gives us insights rightly, immediately. So when we speak about, okay, 1.64% of the confirmed cases are active, that means, okay, it's very minimal. That's where statistics comes into use. So instead of having a lot of numbers, we organize them, we summarize them, and we can represent them in charts and any other figure. So, but what we observe from this graph that very minimum 1.64% of the confirmed cases are active. This was based on the January 7th information. Then almost 98% of the confirmed cases have recovered, so it's very good to know, and only 0.47% have deaths have occurred. So that's I think a very good way of understanding and learning about any case. There's another graph displaying the immunization coverage of India, and this has been taken from the World Health Organization website, and this is a recent information from the website. So if you notice that this, if you have some information, just numbers, it will not be so easy to understand. So if you are summarizing in the form of a graph here, if you see that green denotes, the dark green denotes that 95 to 100% of that particular vaccine has been immunized, right? So the dark red denotes that 50 to 59% has only been immunized. So you can know which of these vaccines have been more have been applied to the normal people of India. So that's how statistics comes into use. So this is just an example here. Okay, so you can go through this again. I will be forwarding you the slides so you can later, because there's a lot of things to say. So I'll just be a little bit more fast. Another example on the standard deviation, the variability which I was talking about. Let us look at an example. This is a study of women's nutrition for women aged 25 to 50 years, and they had taken a sample of 737. The following variables were measured: calcium, iron, protein, vitamin A, and vitamin C. So this example has been taken from an online source from the Pennsylvania University. Let us look at how we do a descriptive analysis here. So we have summarized the information. The data was there. These are the different variables. So that's what we say variables are: calcium, iron, protein, vitamin A, vitamin C. And we have calculated the mean and the standard deviation. So if you notice, the standard deviations here are very high and very close to the means. So if there is a higher standard deviation, there is a higher variability. So what do we mean by that? There's a large amount of spread. The values are quite far away from the central value, the mean, the average. the standard deviations are large relative to the respective means, especially if you see vitamin A and vitamin C. So this indicates a high variability among women in nutrient intake. So that's how we can understand, okay, even though they have taken the supplements, I think there's a lot of variability in that. There's another example to explain you much further about standard deviation or variability. So let us take this small example. Suppose that the Director of Public Safety wants to evaluate 2 ambulance services that have contracted with the city to provide emergency medical aid. As a part of the investigation, she has collected the data that the mean response time to calls. So there are 2 services, we say service A and service B. And the mean response time for the call, attending their call, is 7.4 minutes and 7.6 minutes. So if you look at the means, it says, okay, they're almost the same. By just looking at the means, we just have an idea that, okay, they are almost there. So it's okay to take both the services, but that may not exactly be so. If we know the standard deviation, then we will have a different view. So if this is the spread of the 2 response time of the 2 services. So if you notice in service B, the curve is more flatter, right? So there are more observations towards the low and the high values. So that means there is a higher variability there. But here in service A, the values are more consistent towards the mean. So that means there is less variability. So the higher the standard deviation, the higher the variability. So a lower standard deviation is always good because it's more closer to the mean, because the mean is representing the data. So that's representing the sample. So what can we observe from here? Service B was much more variable in response time and had more scores in the high and low ranges and fewer in the middle. Service A was more consistent in its response time and its scores are more clustered or grouped around the mean. Both distributions have essentially the average response time. They have the same response time, but there is considerably more variation or dispersion. A study of variation is also called measures of dispersion in the response times for service B. So that's very important. So whenever you are doing some summarization, if you are finding a mean, it is necessary that you have to find the standard deviation as well, so that the person who is or whatever, the person or whatever is necessary for the particular research, they will have an idea of what is the amount of spread. Let us now there's a lot of things when we are collecting data, there's sampling procedures, but I'm not going to focus on sampling because sampling, again, it will take one more session. So I will not go to sampling. I will directly move to inferential statistics. That is the testing which we do. So the most common tests to compare numerical data point values are the t-test, analysis of variance, and linear regression. I think you all have heard about p-value. I don't know if you have done some research, you have always come to this. So the p-value is used, so this is actually the probability value, is used when we wish to see how likely a hypothesis is true. So if the hypothesis is true or not depends only on the p-value. Confidence interval, again, I think this is a term which you have come across. Confidence interval are typically used when instead of finding the mean value of a sample, we want to find a range. We just don't find out the mean. We say that we have a range where our true parameter is supposed to belong to that interval. So that is likely to contain the true population value. So when we are doing testing, we do testing on the basis of a sample, but the conclusions are the inferences on the entire population. Hypothesis testing, I'll just give you an overview of how we do hypothesis testing. This is one of the important areas for medical or clinical research. We define the 5 stages when carrying out a hypothesis test. We will first define the null and alternative hypothesis under study. We will collect the relevant data. So data collection is again another area where you have to follow certain rules. You have to follow the sampling procedures. Different sampling procedures are there. You have to calculate the value of the test statistic specific to the null hypothesis. So what kind of test? There are different tests. So it depends on what test you are doing. Then you will compare the value of the test statistics to a known probability distribution. And then you will interpret on the basis of the p-value and give your decision. So let's see how we work on this. I will just focus on one sample t-test. So first, we are going to define the null and alternative hypothesis. There are two hypotheses. It's a statement we are going to claim. So, for example, the mean in the population equals to some value. Say it's mu naught here. 0, we say mu naught or mu 0. Sorry. And there must be another hypothesis against the null hypothesis. We call it the mean in the population does not equal mu naught. Now, what's the null hypothesis? I'll come to that in the next slide. Then collect relevant data. So the next step is to collect. First, we will set up our hypothesis that this is what we are going to test. For example, if you want to say that there are two medicines treating a particular disease, so we will say medicine A is the same as medicine B. So that will be on. We want to check whether it is true or not. So we'll say our null hypothesis is medicine A is equal to medicine B. Okay, I'm just this is a simple example. Then we will collect the patients who are taking the medicines, and we'll try to find out how they are responding to the medicine. So we'll collect relevant data from a sample of individuals. Then after doing that, we will try to calculate the value of the test statistic specific to the null hypothesis. So in this case, we are doing t-test. So t-test is a very popular famous test which is used mostly in the medical and clinical research. And this is not just one test, there are many tests. There are pair t-tests, there are t-tests for difference of mean. So there are different other t-tests. And this t is it's a tool for comparing our null hypothesis for taking decisions. So this is when we have some measurement. So this is that measurement. We will use this. And this t follows a distribution called t distribution. This you will not understand with n-1 degrees of freedom. If I have to make you understanding, I think I have to go to a chapter. But this is a general idea of that. Then we will compare the value of the test statistics. This is what I said now. And for this, we are going to refer to the statistical table. We have different statistical tables. So for t, we are going to refer to the t distribution table. Interpret the p-value. So then we will try to compare our t, which we will calculate, with our statistical table. And then we try to find out the 95% confidence interval. This is the formula for calculating the confidence interval. X bar is the mean. So that is the symbol used. X denotes the variables, and X bar denotes the mean. So mean plus minus the t value, which is derived from the statistical table. And this 0.05 is because of the 95%. So whenever you're performing a test, we have some certain level of significance. We call it the level of significance. So 5% we might commit errors, and the remaining 95% we have to be confident about our parameter. So that's 95%, or the 5% significance level. That's a level of error, 5%. And S is the standard deviation, and N is the number of samples here. So this t is the percentage point of the t distribution, which gives a two-tailed probability. So this is a much more explanation you will need for that, but I'll not go to that now. So let us take, before we done, I'll just give you an example of the null and alternative hypothesis. The null hypothesis is the statement we are going to prove, we are going to claim. So that's a statement of no difference. We assume it to be equal, that medicine A is equal to, so there's no difference. So always we construct our null hypothesis in such a way that it is used for testing for possible rejection, So for example, if we are interested in knowing whether the weights of babies significantly increase in 2-month growth period, so we'll set up our null hypothesis. The weight of babies is not different. So there is no difference in the 2-month growth period. Against the null alternative will be, it is different. So we'll try to possibly, testing is always done to possibly reject our null hypothesis. Okay. So that's how we set up our hypothesis, the null hypothesis and the alternative hypothesis. So using the p-value, we talked about p-value. So how does we reject or accept our hypothesis? So we must make a decision about how much evidence we require to enable us to decide to reject the hypothesis in favor of the alternative. We have to favor the alternative and reject our null hypothesis. The smaller the p-value, the greater the evidence of rejecting the null hypothesis. So what's that smaller? So we have to have a level. So conventionally, we consider that if the p-value is less than 5%, so 0.05, there is sufficient evidence to reject our null hypothesis. So if you have a p-value, if you do the test and you get a p-value which is less than 0.05, 5% is the standard. For higher clinical trials, for more sensitive clinical trials, it should be 1%. That's 99% confidence level should be there, 1% margin of error. So in that case, your p-value should be less than 0.01. As there is only a small chance of the result occurring if the null hypothesis were true. We then reject the null hypothesis and say that the results are significant at 5% level. So in terms of now, this is because the reason it's the COVID-19, right? People are researching. So we are 100% sure that they are taking the confidence level to 99% because there's sensitive cases. So whenever it's higher and said the sensitive medical or clinical cases, the confidence level should be 99% and 1% margin of error. So that's how we use the p-value. And this is an example of finding the confidence interval of a mean. So let us look at an example. We are interested in determining the mean age at first birth in women who have bleeding disorders in a sample of 49 such women. Again, this is an example taken from a source. So it's an old experiment from 1997. So we first find out the mean age of women at birth of child. So that's what I said, X bar is the average. So it was 27.01 years. Standard deviation is, again, we calculate the standard deviation by using the formula. We calculate the standard error, which is S over root n. That's S is the standard deviation, and is the number of samples. That's the sample size. So the 95% confidence intervals, if you go back to the previous slide, I gave you a formula. So we are using that formula. And, that 2.011 is the t-value, and 0.7326 is the S over root n. So we get a confidence interval, 95% confidence interval. So we are 95% certain that the true average, that means true average means the average, this is a sample, but we are talking about the population. So we are talking about the entire population of women who have bleeding disorders while giving birth to a child. So this conclusion is based not on the sample, it's based on the population. So we are 95% certain that the true mean, so true here means about the population, at first birth in women with bleeding disorders. We are 95% confident that this will lie in this interval. And the more the width is narrower in the interval, it's more precise. So if you have a wider interval, the estimate is not as precise, as accurate. So if we repeat this experiment for a number of times, the range of values determined in this way would contain the true population mean, or 95% of. So 95% confident means we have to repeat this experiment a number of times. So that's why we say we are 95% sure that our true population will belong to this interval. So this is just an example of how we construct the confidence interval. Okay, I don't know why it's not going. It's not moving. Again, there's another example. I think I will not go to this slide because this is for a large sample. So when we have a large sample, when the sample size is large, so sample size is large means there's 1,513. In the previous case, it was just 49. So when you have a large sample, your that value changes to 1.96. So we use the Z test. There are different types of tests. So this is another example. But I think since I will be giving you the slides, you can go through this later on. So this is similar to the one, but only the difference is we are using the Z statistic here, not the T. Okay, I will do an example I have just done. This is a fictitious example. I have used R programming, and it is a example based on data available in R. So inbuilt data. So it's about a data called tooth growth, which is it's actually upon the growth of in the response in the length of autonoblast in 60 guinea pigs. Each animal received 1 or 3 dose levels of vitamin C, so in the 3 different milligrams by one of the delivery methods. I wanted to do a t-test, and I just wanted to show you. I used a software programming here. I did not do it manually. So if you notice here, what we did, we write the null hypothesis that the mean length is equal to 16. We assumed that it is 16. Against that the mean length is not equal to 16. I don't know what is the mean of this data. So I have assumed this as null hypothesis is 16. By looking at the data, I just did not calculate it. So we did, we run it in the our programming, and I saw, if you notice that we have the t-value there, that I called it the t-statistic, so that is the t-value. And the p-value. So what kind of conclusion will you give here? If you notice that this is less than 0.05, 5%, so that means we have sufficient evidence to reject our null hypothesis, right? Because it is less than 0.05. And also it's because I'm using a software, I don't have to do anything manually. I also have the confidence interval, that's the 2 values. So this is the true population, the mean length of the population of the pigs is supposed to lie in this interval. So we are 95% sure that the true average, or the average length of the guinea pigs is going to belong to this interval. And also, if the true mean is given there, and if you notice below, that is the true mean. So it's this, that means we have sufficient evidence to reject our null hypothesis. So that's how we do. This is a small test. This is just an example. It's taken from our database. So you can do it later on for any other health or clinical data. So another example is there. I can't see from here, but this is a anyway, this is a test for one sample test, again done for triglyceride levels. So they have done a t-test, but this has been done manually. So what they did is they compared the t-value with the statistical value here, and they have rejected the null hypothesis. But in the earlier programming, because earlier we were doing it manually, we had to refer to the statistical table. So this is just an example again for doing a one sample test. So because I cannot see it much better, and it's a small, no problem. You can refer. This is the same way I have done this example. So, now the graphs of these two. The two histograms are given. If you notice, the data, the original data is the first graph. And if you notice this, the most of the values are going towards the right. So that's a skewed curve. So whenever you have some values either more towards the right or more towards the left, we call it skewed. So while doing testing, you have to keep in mind the data should not be skewed. It should be a normal symmetrical curve. So when it's not normal, you have to make it normal by using the logarithm. So and sometimes when even if it's not normal, then you have to use non-parametric tests. So, but here I'm going to do when we change the data to logarithm, we saw that it's going to be asymmetrical, almost asymmetrical. So symmetrical means it's following a certain distribution called the Gaussian or the normal distribution. So then you can perform all kinds of testing, parametric tests like t-test, z-test, any other test you can do. But when it's not normal and any conditions, then you have to go for non-parametric tests. There are different tests for non-parametric as well. So this again has been taken from a bulletin, which was done manually. These are the statistical table. So we refer to this t values are there, the z values, the chi-square for qualitative data. If you're testing something qualitative, chi-square. So this is just an example of a t-test, testing of hypothesis. There are many other tests which I cannot cover today, but definitely in future, we hope we can take certain areas and we can dedicate more time to that. Then another part, which is also included sometimes in inferential statistics, is the prediction, where we do regression and time series modeling. So this type of models like multivariate regression, ARIMA, that is auto-regressive integrated moving average model. And these type of models we do. There are many more models which I will discuss now. For some examples for doing predictive statistics is predicting continued demand for radiology services in a medical center, predicting pneumonia risk and hospital 30-day permission. Then we cannot do testing of hypothesis. We have to do some prediction statistics. So in that case, we have to either use regression modeling or we have to use time series modeling. So it depends on what kind of study you are going to do. So you have to be rightfully judge what kind of statistics you need to use to design your research. The first one I have done, it's a project under me, which I have done with some of my fellow faculty members there. This is the abstract of, there were 6 students who did this. So this was which I have done. There were different, one did regression modeling, another student did time series modeling, another student, James, he did also queuing theory, which is, I have a specialization in queuing theory. So I have used queue modeling also here. So this got a lot of appreciation. It was very recorded well. So this is one of the way you can, if you feel like modeling, you can go for time series or regression. So just to say finally that the involvement of statistics with different perspectives. So there's different. This is just a tiny bit which I have spoken. I'm given you. I've just given you a brief overview. There are a lot of things to know. There are a lot of things to discuss. You can keep in touch with me if you want. If you want any kind of help. But what I would like to say finally that as it has progressed, research has progressed, health sector, clinical sector has progressed, a lot of advancements and methods have come across in statistics. So there is a lot of, it has widened. Statistical methodologies have widened. So we are starting to use some generalized linear models. We use multiple linear regression. We are using survival analysis, categorical data analysis. These are more complicated spatial statistics and Bayesian methods. So they are mostly nowadays, if you see research in Western countries, they have started using more complicated and sophisticated statistical techniques. So if we are doing something manually, and we have been doing it manually, there is some room left for error, human error, right? So conclusions may become dangerous, especially in the health sector. So nowadays, there are a lot of software as accurate results. So it's better if we can use some software. So the most popular softwares for doing health and clinical research, you can go for IBM Statistical Package for Social Sciences. This is very popular, SPSS, but I think this is not free. R programming is free. You can download it from the R CRAN website. SAS, Minitab, Stata, I think they are also, you have to buy those softwares. And Excel, I think everybody knows about that. But you can use R, which is very convenient and very easy to use. Even SPSS, but SPSS is not free. So whenever you do research nowadays, you do not have to do anything manually. You can go ahead with the different statistical programmings. And anybody who would like to collaborate with me, would like to do projects there, or if any help related to any statistical matter of your research or anything else, my research are queuing theory, biostatistics, regression modeling, and you can email me in my email ID. So thank you very much again. I will leave it for the question and answers. If you have any questions, you can ask me. So any questions? About the, again, you want it again? Okay.
Yes, ma'am. Is it specified
Sorry? Yeah, it's not, you cannot say a direct answer is error-free result. This is always, there will be some amount of error. right? You cannot be 100% confident. So we have a margin of error that's 5% mainly for the standard. But if it's more medical cases, higher medical sensitive cases, you have to have your confidence level of 99%, 1% margin of error. So confidence intervals is not that I'm 95% confident. No, you repeat the experiment a number of times. The same experiment, you repeat a different number of samples, and then you will see that 95% of the times that average, when we take a sample, we have a population, you have different samples. You take 10 samples from here, 10 samples from 10 samples. So you have to, when you see that the number of times you're repeating, that the true average, the average of the sample is belonging to that interval. For this next sample again, the average belongs to the interval. Again, so that means if you again keep on repeating, so we don't keep on repeating, but nowadays there are softwares to keep on repeat the sample. So that means we are 95% confident that our true population parameter is supposed to belong to that interval. It's not error free. It's not error SPSS is very good. There are very clear steps. In R, you have to learn a lot of coding languages. So convenience-wise, SPSS is good, but it's not free. I don't know whether sometimes the institution gives it. If the institution have it, then it's fine. Otherwise, you can go for R programming. There are many books available on R, so you can also read the books and do. But coding is main important in R.
It was a very nice presentation for all the data present for my project about this. I just wanted to ask, till now in our thesis, whatever we have been writing, we have included this key level of study. Any other method is there from which we can see the confidence level in our experiments, which we may include in our thesis?
As far as you're doing testing, we always have the p-value. If you're not doing,
it is the
better way. Okay. If you're doing testing, if there's if you're doing any other kind of research, then it will depend on what you're trying to do research. But testing, it's always the p-value. Yeah.
Is there any other percentage that we can set for the p-value? Because when I have been working with the software graphic, there was a option to change the percentage values.
It depends on the null or the alternative hypothesis. So if you, like here, I took the alternative hypothesis as a not equal to, so there might be, so I said the average is 20, but the average may not be equal. So it can be either less or it can be either more. So it depends on the alternate. So when we are choosing the test, there are three types actually. There is one-sided test, two-sided test, and there are two types of tests: right, left, and two-sided test. So there are three tests total. So it depends on the type of test you're using. So it may change. So p-value, but the standard is 0.05 and 0.01. You can get in, yeah. It's open. It's an open resource. Yeah. But you have to know a lot of coding. That is the only problem. But books are available now. There are a lot of YouTube videos also. Yeah. Okay. Okay. Thank you so much, and I'm very grateful to the NEMCARE group. Yeah. Okay. He has a question.
If our faculty is those are doctorate, from our side, if we want to send them for first doctorate program for 6 months, how the university can be help our faculty to go as a postdoctoral?
Actually, even I have not, still now, I don't know the procedure, but if you really have a thought in your mind that you want to send them for a postdoctoral program or some kind of certificate program, I can discuss this with our department or our, if it's necessary, we have a biological department. I can discuss.
Yeah, we can send for 1 month
refresher. So it will be a good idea. It's a very good idea. In fact, I can carry forward this information and I can discuss with our department and say if there is any kind of collaboration. Yeah. So I think their biostatistics department can help in that. Yeah, I can maybe coordinate for that if you wish in future. And I also recommend, just want to give you a suggestion that I think there should be a course of biostatistics, because if any master's level, yeah, you have a paper. So there is faculty of biostatistics. Yeah, yeah, that's fine. Yeah, because in one day or 1 hour, nobody can master statistics, so they might need the help.
Because we have already mentioned
Yeah. Yes.
In that they mentioned that their professional skills, their learning process is quite different.
Quite different.
Quite
Yeah.
different from Indian lecturers.
Yeah.
And Indian so if we can send our teacher to learn this thing from great university, so it will be a great opportunity for.
It's a very nice idea, and I think I will take this forward. I would like to talk with my organization, especially my department first, and then I will see what are the possible ways of doing that. Yeah, if there's a possibility. I don't know what's the might be some source of funding, or definitely we can take this ahead. Yeah, thank you. Yes.
This method you can provide any overseas project, or like as you're working in UK, so the university will have provided overseas project, or is it such kind of
project? Well, I don't know actually about how your projects are carried out. We have master's degree projects because every student is supposed to have a project there. They will have 2 or 3 modules, and modules means courses, and then they have to go for a project. It's mandatory, compulsory. So I don't know if there is a project here for the master's level students, because I don't know what's how they are being run here. So if you have a personal intention of doing any project, yeah, you can contact. I have given you my email, so you can just talk to me that I'm a student or I'm a faculty here, and we want to do a project, and we can collaborate. Anytime you can just email me. That's fine. Any more? Okay, then thank you very much, and I'm very grateful to actually. I wanted to say this to Bikramjit, my nephew. He was the first one who initiated that. Why don't you give a talk here? And that he made it happen with the help of Dr. Bhargav and Dr. Apurva, and Nilutpal is there, who's the coordinator. Thank you so much for coordinating. And I'm also grateful to the NEMCARE directors, Dr. Hitesh Baruah, Dr. Mihir Baruah, and Dr. Hitesh Deka. He's not there, I think, so I'm very much honored, and it's my pleasure to be here again. Thank you very much. Oh, this one,
Thank you very much, madam, for such a wonderful interactive session. And definitely it will help us, all of us actually, in our near future for any research work. So now I would like to request our organizing secretary, Dr. Apurva Talukdar sir, to please offer vote of thanks. Sir.
Okay. Thank you, Deepsika, for giving me the opportunity to deliver the vote of thanks. So at the very beginning, I would like to request our Nalini Sharma sir to hand over the token of appreciation to our chief guest, Dr. Monita Baruah ma'am. Thank you, ma'am. Okay. At the very beginning, I should thank COVID-19 because of whom we are lucky enough to have Monita ma'am among us. Otherwise, it was difficult to have a eminent personality like ma'am from UK to deliver a lecture physically here. So thanks to COVID-19. So on behalf of NEMCARE Group of Institute and on behalf of ASTU also, I would like to give my heartfelt thanks to our chief guest, Dr. Monita Baruah ma'am, for giving her valuable time and spending her time with the topic biostatistics and statistical analysis in terms of faculty development program. So, and I would also like to request our guest of honor, Dr. B.R. Phukan sir, academic registrar, who just left the event due to some other engagement because some exam is still going on in our campus itself. Also, I would like to thank our principal sir, Dr. Bhargavjyoti Souria, who has supported to organize this event smoothly in a very short period of time, in a very short notice of time, because last week only we have discussed regarding this, and we have made arrangement all these things in a very quick sort of period of time. And I would like to thank our trustee member, Dr. Monita Baruah ma'am. Her presence really enlightened us because she has traveled from Guwahati to just to listen this FDP. And finally, I'd like to thank all my colleagues, all the faculty members, colleague faculty members, especially Nilutpal and other faculty members who has worked hard to organize this event and to make it happen, because we know all the faculty members are so busy in terms of exams and other engagement, but still they have managed their valuable time to organize this event smoothly. And finally, I would like to thank all the participants who are present today, and some participants are online also, because we are going online through Google Meet. So some members are there who are listening online. So I would like to thank all of them. So lastly, I'll thank all of you to be present here and spare your valuable time. Thank you. Thank you all.
Thank you very much. Thank you very much. It's been an honor to be among all the accomplished individuals. So at last, I would like to thank all the participants and also the eminent guests and our principal sir and Malini sir and everybody actually for giving us your valuable time. And lastly, I want to tell that we will take a picture, group picture actually. So after having tea, actually we have arranged a high tea. So I would like to tell that everybody please proceed for the high tea, and then we will click a photo. Thank you everybody. Thank you very much.